Welcome to this lesson on dichotomous keys. In this session, we'll explore what they are, their advantages and limitations, and how to use different types of dichotomous keys. Before we dive into the lesson, let's go over what you'll learn by the end of this lesson. You will be able to define taxonomy keys concept, especially dichotomous keys, to compare the pros and cons of using taxonomy keys in the classification of organisms, to understand and be able to construct bracketed and indented keys in dichotomous key formats. Taxonomy is a branch of science that provides a robust and accurate way to identify organisms. This identification process is scientifically carried out using a taxonomic key. Keys are generally used to identify specimens using either structured keys, also known as single-access keys, and polyclave keys, also known as multi-access keys. The taxonomic key was historically used circa the 17th and 18th centuries and became popular during the 19th and 20th centuries. Taxonomic keys are built upon the evolutionary relationships among organisms, and their construction mirrors these connections. When a taxonomist discovers a new organism or group of organisms, or acquires new information about a known organism, the findings are published in scientific journals and integrated into the revision of the corresponding taxonomic key. This ongoing process ensures that biologists have access to up-to-date and accurate keys for their work. Taxonomic key, also known as diagnostic key, determinator, or artificial key, was used for at least two biological purposes. 1. Recognition of major divisions within groups. 2. Detailed identification of taxa to genus or species name. The dichotomous keys are usually heavily illustrated and designed to facilitate users with minimal background with the systematics of the group studied. As for the application, the keys were extensively developed in plant taxonomy and entomology, where a group of related taxa can be very large, and field guides often included keys to orders, families, and genera. However, there are a few drawbacks to using taxonomy keys, such as 1. It does not work if the species to which the unknown belongs is not included in the key. 2. It may lead to misidentification if an unknown shares some of the attributes of species that are already included in the keys. 3. The specimen is a hybrid between two species, especially in plants. 4. The species is fragmentary or lacks characters needed to follow the key. 5. The specimen falls on the borderline of a character supposedly separating two species. Despite that, keys have been proven to work perfectly under the right circumstances. Even long keys, or up to 200 couples, rarely require the user to make more than a dozen choices to get to any given species. A key character in taxonomy study is the one that provides some clearly and conveniently observable distinguishing feature that helps identify an organism. In fact, good keys will lead the user to the desired results with minimal identification steps. However, a character useful for the scientist might be obscure to the reader with minimal taxonomy knowledge. Instead, in a high taxonomic rank, the choice of characters may be very difficult because some exceptions are likely to occur with most characters. Hence, choosing the best characters to suit the taxonomy keys is crucial. Here are some guidelines for choosing the best characters in taxonomy keys. In this session, we will cover only structured keys, also known as single-access keys. One of the renowned single-access keys is the dichotomous key. Dichotomous means dividing into pairs. The dichotomous keys consist of couplets and leads. Each statement leads to another pair of choices, hence narrowing the search until the final statement identifies the organism in question. Good dichotomous keys have several characteristics, such as 1. Each couplet is composed of strictly contrasting leads of 2 or 3 contrasts. 2. The style is telegraphic. 3. The characters are readily observable if at all possible. 4. Character contrasts should not call for value judgments, for example, lighter or darker without reference to another shade on the same organism. 5. The key should be clearly labelled. For example, it is applicable to particular groups such as ages or sexes. 6. Characters that are apt to rely on expert judgment should be illustrated. There are 2 main formats for constructing a dichotomous key. The first is the bracketed or open key. A bracketed dichotomous key is the most common and practical type. It is also one of the easiest for beginners to understand. In this format, the 2 leads of each couplet are placed side by side, and each choice tells you which number to move to next. This structure allows you to clearly compare contrasting options before moving forward. Example 1 shows how a bracketed key can be used to classify animals into major groups such as fishes, amphibians, reptiles, birds, and mammals. We begin at step 1. If the organism has fins or no appendages and lives in water, we move to step 2. If it has legs, wings, or flippers instead of fins, we move to step 3. At step 2, fishes are further separated into 3 classes. If instead we followed step 3, we enter the terrestrial vertebrates. Finally, step 4 separates the remaining groups. Notice how each step provides 2 opposing descriptions. By carefully choosing the correct one, we gradually narrow down the possibilities until we reach the correct group. Example 2 is another bracketed key, this time for bird species in the genus Tyrannus. Step 1. If the bird has a white belly, go to step 2. If it has a yellow belly, go to step 3. At step 2, if the tail has a white tip, the species is T. tyrannus. If the tail has no white tip, it is T. dominicensis. At step 3, if the exposed culmen, the upper part of the bill, is equal to or shorter than the tarsus, the species is T. melancholicus. Otherwise, we proceed to step 4. At step 4, the wings are compared. If the outer primary feather is shorter than the 6th, the species is T. voliferus. If not, it is T. verticalis. Again, notice that each step is presented as a pair of bracketed alternatives. The observer must read both carefully and decide which matches the specimen. This ensures a clear and logical pathway to identification. The second format of a dichotomous key is the indented or closed key. In this format, each choice is indented beneath the one that came before. This creates a tree-like structure where the pathway is visually clear. Unlike the bracketed key, you don't jump back and forth between numbers. Instead, you simply follow the indentation downward. Indented keys are helpful for short and simple classifications because the logical pattern of the choices is very easy to see. However, they are less practical for long or complex keys, as the constant indenting can push the text too far to the right, making it harder to read and compare options. Example 3 shows an indented key for bird species in the genus Tyrannus. We begin at the top. If the bird has a white belly, the key indents us to the next choice. If the tail has a white tip, it is T. tyrannus. If the tail has no white tip, it is T. dominicensis. If instead the bird has a yellow belly, we follow the other path. We then compare the beak and leg. If the exposed culmen, or upper part of the bill, is equal to or shorter than the tarsus, the species is T. melancholicus. If not, we continue further. At this deeper indentation, wing features are compared. If the outer primary feather is shorter than the 6th feather, the species is T. voliferus. Otherwise, it is T. verticalis. Notice how in the indented format, You can visually follow the choices straight down the page without needing to jump to another numbered step. This makes indented keys easier to navigate for short examples, but in long and detailed keys, users might lose track of where they are. That's why indented keys are best suited for smaller groups, while bracketed keys are often used for larger or more complex classifications. In conclusion, taxonomic keys play a crucial role in the identification and classification of organisms, offering a structured and efficient approach to understanding biodiversity. By categorizing organisms based on observable characteristics, taxonomic keys provide a systematic method for identifying species, from broad taxonomic groups down to specific genus and species.